Good morning to all of you, and at the outset I'd like to thank the university and the Indo-France collaboration for asking me to speak here. Very thankful note to Dr. Professor Vengama, who's somewhat like my mentor. I'll be talking on the gut microbiome, and uh, we will focus on neurological disorders because that's the area I represent. Um, we are what we eat, so much so, what we think and what we behave is also what we eat. And just who said this? So it was Jean Anthelme Brilia Savarin. I don't know if I pronounced it right, but that's a French person. So obviously, this talk is very, very, appropriate in an Indo-French forum. But even before it was said by the French, the Indian Sanskrit philosophers had clearly stated that the state of the mind and behavior depends on what you eat. And our food was divided into three categories: sattvic, rajasic, and tamasic. I hope all the youngsters can read about these three forms of food and how they influence our brain and behavior. It's been millions of years and our diet has been changing, and we've heard of these changing diets. We've heard of the paleo diet, the neo diet, and then you have evolving mind and DASH and keto diet, and all these have come up as we have moved on in our development as humans. What is intriguing is that the number of human cells is about 30 trillions, but the number of bacterial cells is 38 trillion. So obviously the bacteria are trumping the human cells, and we haven't even realized it. We are at the tip of the iceberg. We haven't even let it sink in as to how much these bacteria influence these human cells, which are lesser in number than them. So if you look at the species of bacteria alone, the gut is a melting pot for bacteria. You compare it to the skin. The skin has just 10 to the power of 11 bacteria on it. But you look at each part of the gut. You look at the oral cavity, that's 10 to the power of 12, duodenum, colon, ileum, jejunum, stomach, all of them put together have so many 10 to the power of whatever. So the gut has many more times the bacteria that any part of your body can have. Okay, so that's very, very important to remember. The size of the organism does not matter. The size does not matter. The number does. We'll go through in this talk. So let's look at the, gut microbiota. Everything has a classification, and there's a typical microbial classification scheme here. And for some reason, I've forgotten my reading glasses in my laptop bag. If one of the girls here can be kind enough to get me my reading glasses in my laptop bag, yeah. So 90% of the gut bacteria belong to the phyla of Firmicutes and Bacteroidetes. So this is for the youngsters here. You must memorize this. Yeah, it's there in that, yeah. So there are several factors which influence the gut microbiota, and you can see here, this is a typical, thank you. This is a typical Indian gut microbiome, and as you can see, all the French people here would have realized that they're visiting South India, which is distinctly different from North India. So the gut microbiota in different parts of India is also different because our food styles are different. Believe me, my husband is from Andhra, so I know different food styles. I make South Indian breakfast in the morning and have North Indian parathas in the afternoon. So the bacteria are different. The gut microbiome is different. North Central India, the plant-based, which is predominantly the Central India, which is Madhya Pradesh, mostly thought to be vegetarian or plant-based, is Prevotella and branch-chain amino acids, as you can see there. So the BCAA and LPS biosynthesis pathways are enriched here. And Southern India, where it's omnivorous, vegetarian plus non-vegetarian, as we can see in the food habits here, are mostly Bacteroidetes and short-chain fatty acids transporters enriched. So you do have a regional variation in the Indian gut microbiome also. And the evolution of the human gut microbiome research is not new. It's, the Dutch who discovered something. It's got something to do with the microbiomes in the gut, because he first observed more than 1,000 microbes in his own stool. So you can imagine what a researcher he was. He was researching his own stool. And then secondly, in 1842, it was the Scottish person, John Goodsir, who then described the gut bacteria Sarcina ventriculi from a patient. And when the Scot are ahead, the British can't be far behind. So the British physician Lionel Smith described microbes in human stomach, intestine and stools, and so the germ theory of disease got well established. And to cut it short, as late as 2011, the gut microbiota regulate brain development and behavior is what has been proposed, hypothesized and researched. It's still, in process, as the previous speaker highlighted, but there are seminal studies which establish that the gut microbiota has a great role in brain development. So how do they influence each other? In fact, the gut is called the little brain. The stomach is called the little brain. So look at this diagram, and this is the gut, and this is of course the brain. So you have microbiota derived influences and host derived influences of this interaction. And the microbiota derived influences range from these, that is amino acids, short chain fatty acids, bile acids, vagal nerve effects, lipopolysaccharides, neurotransmitters. We know we are an interplay of neurotransmitters. Our mood and behavior is the interplay of neurotransmitters, gut hormones and immune functions, which are dependent on many of these processes. And the host derived, the hypothalamic pituitary adrenal axis, will affect the gut motility, gut permeability, immune functions and mucus secretions. So it's a bidirectional relationship. Nobody exists in isolation. We all exist because of our networks, because of our interactions. The previous speaker has already highlighted the role in Parkinson's disease, and believe me, we are still exploring that. We know that there's a whole theory of how Parkinson's disease starts in the gut. So let's shift. I'm not going to touch upon Parkinson's because it's been spoken about. So let's move to Alzheimer's, which is another area of my research, because I do a lot of cognitive research also. What has been found is a low abundance of these kind of bacteria, Ruminococcus C, Tursi, Bacteriaceae and Clostridiceae, and high levels of microbiota-derived metabolites, TMAO, in the CSF of AD patients. So this is a 2018 study. And low levels of microbiota-derived SCFAs, which is a 2022 study, which decreased the BDNF, decreased, the P13K and AKT signaling, was associated with high A beta levels, microglial activation and blood-brain barrier disruption, leading to neuronal death, cognitive impairment and dementia. So this is one of the theories of a very complex disease like Alzheimer's. Alzheimer's is not a simple disease for any researcher to decipher. The fact that we haven't established a cure indicates that there's more research to be done on the gut microbiome and Alzheimer's. Let's look at multiple sclerosis. This is a chronic disease with episodic manifestations, affects the young people, particularly women. So what has been found is autoreactive peripheral CD4 T cells migrate into the CNS and initiate the MS process. Probiotic supplementation showed an improvement in disability scores, depression, and inflammatory markers. This is 2017 research. Clostridium perfringens type B toxin could play an important role as a nascent lesion formation leading to ability to bind to oligodendrocytes and myelin, and increased, I find these pronunciations very difficult, Akkermansia and some decreased Bacteroides showed low SCFA and inflammation promotion, leading to demyelination, cognitive and motor dysfunction in multiple sclerosis. There's a huge role of diet in multiple sclerosis. In fact, there's a theory of an American neuroscientist who says avoid non-vegetarian food and milk products, any animal products, and multiple sclerosis will come down and the relapses will come down. So there's a huge role to play in many of these diseases. So to give it in a nutshell, there's a role in Alzheimer's, Parkinson's, multiple sclerosis, and it works through causing the gut microbiome dysbiosis, which then triggers off inner cogwheel mechanism, various downstream cascade pathways which work on gut permeability, SCFAs, LPS, cytokines, blood-brain barrier dysfunction, leading to neuroinflammation and damage into the brain, and an irreversible cascade which then follows. My interest is, of course, epilepsy, one of my interests. And let's look at epilepsy. It's in 1907 that epileptic seizures were thought to be arising due to poor dietary habits, and toxins in the gut could reach the brain cortex to cause seizures was one theory, and this theory was given by Connolly in 1907. In 1910, there was intestinal toxemia theory and constipation, which causes direct idiopathic and peripheral epilepsy for whatever that was in those days. And then in 1916, Charles, interestingly found spore-bearing motile organisms in the blood. Now, this is very amusing to us right now. However, this theory was then, did not confirm these bacilli in the blood. And of course, the gut theory was thrown out for epilepsy. However, the first modern evidence of epilepsy gut connection came in from the diet. We know there are specific diets which cause a good response in epilepsy. We have published two this year in Green Journal Neurology with an impact factor of 11, showing how ketogenic diet and modified Atkins diet helps even adolescents to reduce seizures in 30% and control seizures in 9%. So what has been done in this study was that drug-resistant epilepsy, which were 14 in number, and healthy control, which were double in number, 30 in number, they had a 1 week of ketogenic diet, and they underwent RNA sequencing, and 64% of these were responders. And you can see here the gut microbiota differed in the DRE patients compared to the healthy control, but the shortcoming of the study was there was no pre and post KD analysis of this data. So there was a difference, and there was a decreased alpha diversity in the DRE. We'll come to learn what is alpha diversity. There's another study here where here you can see ketogenic diet has been used, which has been used, was the first study to show that in mouse models, 2 mouse models, as we heard of mouse models in the previous talk, that is, there was decreased alpha diversity, increased akkermansia, increased parabacteriosidus, increased hippocampal GABA levels relative to glutamate, protects against seizures. So that's how the gut microbiome theory for the efficacy of ketogenic diet came into play. Following this, there are several studies. There you can see the list of the studies. I don't want you to read through it, but the gist of all these studies is that the possible reasons are that the conflicting results, that yes, it has a role versus no, it has no role, is that the populations were mixed. There were various etiologies of epilepsies mixed. There was a huge range, age variation. There were multiple anti-seizure medicines which could have modified the effect of the study of the gut microbiota. There were varied diets of these people in these studies, varied ethnicities. So essentially, it was a heterogeneous data which may have led to conflicting results of the role of the gut microbiome. So what are we doing at our Department of Neurology? I have a PhD student who is funded by Indian Council of Medical Research, who is using whole genome shotgun sequencing on our patients who are undergoing surgery for drug-resistant epilepsy, that is one group, and our patients who are on ketogenic diet, pre-diet and post-diet for drug-resistant epilepsy, and these people who are on ketogenic diet are not candidates for epilepsy surgery. So we have these 2 cohorts which we are sampling. It's an ongoing analysis. We have a sample size which has been derived, and we have age and gender matched healthy control. We have stringent exclusion and inclusion criteria, and the DRE definition is as per the ILE guidelines. We have excluded those with any of these conditions like diabetes, other autoimmune conditions, antibiotic intake recent to the inclusion, because that can modify the gut microbiome, history of other neurological disorders, etc. So what we're doing is we're collecting. It's a very cumbersome process. Only Krish can do it. He is my PhD student who's doing this. So he does a fecal sample collection, he does the extraction, he does a library preparation, raw data, pre-processing, then taxonomic classification, genomic assembly, gene annotation, and all this is ongoing. So this is the results preliminary, and you can see here in the drug-resistant epilepsy patients, there's an increased Firmicutes Acinetobacter, decreased Bacteroides, and this is consistent with Peng and Z's data. This is the results he's penned up over here in the man with knee. And then he's done a taxonomic distribution of the 4 most abundant phyla, which is the Acinetobacter, Bacteroides, Firmicutes, and Proteobacter. And you can see here there's a vast striking difference in the color coding. As you can see here, the healthy control, which were 23 in number, the most abundant phylum was the red, which is Bacteroidetes, and the drug resistant sample, the most is the Firmicutes. And of course, there's a linear discrimination analysis. And here we find, this is what we are finding, increase these, and in the resistant, this is in the control, in the resistant it is this. So basically we are looking at all these interactions, the cytokine receptor, peptidase, ubiquitin, mitochondrial, and in the healthy control we are finding these things. So hopefully the sample size will increase. This is the alpha diversity of our patients, as you can see here. So what exactly is alpha diversity? Note that even though both groups, that is group 1 and group 2, have 4 types of bacteria in richness, the species are more uniformly or even or relative abundance present in group 1 as compared to group 2. So this is the alpha diversity index, which is calculated, which is known as the Shannon diversity index, which he has calculated for our patients. And the alpha diversity was increased in the drug resistant epilepsy groups. The beta diversity, as you can see here, it shows the difference. So common is that which is present in both groups, group 1 and group 2. Unique to group 1, you can see there are certain colors which are unique to group 1, and unique to group 2, there are certain colors which are unique to group 2. So there is a distinct clustering of samples which suggests that the gut microbiome of healthy controls versus drug resistant significantly differ from each other. To summarize, drug resistant epilepsy gut microbiome differs from healthy control. So there is a dysbiosis. There's an increased abundance of a particular bacterium, as you can see here, Bifidobacterium longum in the DRE group, compared to the healthy controls where you have the Privatola copri in the healthy controls. And there's an increase, compared to the healthy controls, there's an increased alpha diversity compared to the DRE group. Both the healthy control and the DRE group possess distinct gut microbiome, and enrichment of butyrate, which is a short chain fatty acid metabolism pathway, is seen in the healthy control group. And why is this exciting? Because you can modify the gut. It's so easy to modify. You can develop molecules. This is an era of, taking up discovery of new substances which can modify the gut microbiome so that they can regulate epilepsy, so that they can modulate seizures. And that's why this work is very, very exciting, and we look forward to, the final publication of this data with more samples. With this, I'd like to thank you again. Let us conquer the demon, that is epilepsy, and let us try to conquer the most complex field of neurodegenerative disorders like Alzheimer's, Parkinson's, and multiple sclerosis. Thank you.
Long live India and France friendship.